Next we have Amaya Viros. Yeah. Amaya did her clinical training in Spain and then was lucky enough to go to UCSF for a number of years, then came back from California Sunshine to eventually Manchester Rain, where she became a Wellcome Clinical Scientist Fellow. In that time, she's also won a couple of awards, including one as the European Society for Dermatological Research Rising Star, which is good. And Amya is going to talk now about how sex and age shape skin cancer. Great, Amya, thank you.
Thank you very much for the invitation today and the opportunity to share the data today. I don't know why this has gone dark. Is there anything I can, you can see it, I guess, but I don't know why it's all dark. It's not my slides anymore. Is there anything I can do from here? Oh, well. I can do this without slides. Nothing? Do you what? I'll wait.
The projector just went. They're trying to fix it.
Oh, good. I thought it was me. I'm so fortunate with these things. Well. Easiest ride of my life. I can actually start if you guys want.
Okay, so it's going to show you the slides. There we go. Fingers crossed. Okay, good. Okay.
Let's see. So I wanted to discuss how age and sex influence the molecular subtypes of melanoma, and my aim today is to, make sure we all identify the high-risk population we have, because we are identifying as physicians and scientists the wrong population that's dying of skin cancer. And there could be a lot more done from the genomics and stratification point of view to help the population. So it's again very dark, but you can see here this is CRUK data showing incidence and mortality. And oh boy, it's going on its own, and it's very, can you pump up the light? So as you can see here, the top graph is a CRUK chart with light blue and pink bars telling you that the incidence of melanoma primarily affects those who are old. The dotted line, which is actually red, marks the 50 age group, and you can already see, if you compare the young group in the incidence and mortality charts, that there's an overwhelming death proportion that occurs in those who are 50 and older. So we can already see that although a lot has been done from the marketing point of view to find patients who have early melanoma, who are young and have early melanoma, we're actually missing to save those who are old. And this is what I want to imprint on you. It's a shame we don't have color, because this lady is a redhead with a bright sunburn. And this is what you usually think of when you think of the usual melanoma patient. And this has been used as a poster child to, funding for melanoma research. And this is what we all have in mind. And as a dermatologist, these are the patients that are most often referred by GPs or by hairdressers, or anybody will send patients because that's what's advertised. However, I wanted to point out the unusual patient, and I wanted to say these are the ones who die. And it's related to chronic sun damage. And I wanted to show you that this gentleman is very dark, and I'm going to be referring throughout my talk to these phenotypes as the British tourist, the young people who go to Spain and sunburn, who are very fair, and the Greek farmers. And I just want to tell you that the death rate in Southern Europe, Italy, Spain, and Greece, is almost as high as the UK. It's only that there's a bimodal curve of death in comparison, so you have more dead that are aged in Southern Europe versus the UK. But the death rate is practically the same, and we're missing these in the UK. So I'm going to hopefully convince you to stratify by age and sex. This is just Cancer Genome Atlas data showing you that when you take metastatic or primary melanoma tissue and you look at age as a survival marker, it is the single most robust predictor of outcome, independently of any markers in the primary tissue. So when you normalize for predictors of outcome to tumors, age will have an unexplained rate of death in the elderly, which is about 5% higher for every 5 years after 60. So by the time you're 80, you have a 20% higher risk of death that's unexplained at the moment. So that's what my lab does. This is the melanoma can arise over the skin of chronically sun-exposed skin, head and neck, Greek farmer, versus the trunk, British tourist, to simplify. But don't forget there are many other minor less frequent subtypes. For example, we have acromelanomas that appear in equal numbers across all ethnicities, so we know it's not just a white person disease. We have uveal melanoma, again, ubiquitous in all ethnicities. We have mucosal melanomas. These are very deadly, and the theory is that we detect them very late, but I do think they have unique pathophysiology to drive very aggressive disease. We also have rare subtypes of cutaneous disease that I won't discuss today. But I just wanted to show you with this picture that's invisible that it can be completely apigmented, and these are the likely ones we miss in the age population. Briefly, just to correlate epidemiological and clinical types with molecular biology, I'm going to call this the Greek farmer model, which is related to chronic sun exposure, the British tourist, and the rare subtypes with very little role or involvement of sun. The Greek farmers tend to have very numerous other UV-related cancers, like BCCs and squamous cell carcinomas, but very few nevi counts, in contrast to the young population, which actually harbors MC1R variants. They're redheads, photosensitive. The picture image you have in your mind of someone at high risk for melanoma, that's what they look like, very different to the Greek farmer. This is the epidemiology, and this is what they tell you. They burned in youth. They have tons of nevi in their backs. And they do correlate with certain molecular subtypes. So we're currently only using molecular subtypes to stratify and guide disease therapy, which is fantastic already, but I'd like to propose to take this further. So if you look at the signature 7 mutations, which are simply a readout of how many mutations UV damage has done to your tumor, you see that with increasing age, as you can see here, the number of mutations driven by UV light in tumors, and this is Cancer Genome Atlas data, and these points are the median for every age group, tends to rise, showing you that as the longer you live, the more mutations your tumor will have because you've exposed longer to UV. So that's great. So of course, these are the ones who are dying. So the question I've asked in my lab is, do UV-induced mutations kill the patients ultimately? Is this why they're dying? Photodamaged skin varies as you age, so of course you have more chronic sun damage on the head and neck versus your bum, because that's what you're hopefully exposing more every day in your life. So we do know that there are different numbers of damage and damage mutations, so UV mutations in perfectly normal tissue, like the eyelids, and this was proven by Íñigo Martín Corena's group at Wellcome, and that accumulates during age. And of course this translates to certain changes in the homeostatic tissue. So what we see and wanted to show you in the pathology tissue is a great amount of solar elastosis or sun damage, which is the degradation of the extracellular matrix that is driven by UV light. So I will show you pathology depicting how very photodamaged head and neck skin looks like. So essentially you've lost all the tissue architecture of the dermis, so you no longer see the collagen fibers. There's a paucity of cells. It's very paucicellular. There's a decrease in number of fibroblasts and in immune cells. And of course, you just see blobs of, there you go, blobs. That's solar elastosis. So that's extrinsic damaged skin, photoaging, so due to extrinsic UV light. And this is essentially the covered buttock, right, of an equally aged person, which preserves all the collagen, as you can see here. The pink is the collagen. As I told you already, the mutations accumulate, and this has been beautifully proven by multiple groups, mainly at Sanger. So the way I try to ask this question is, what happens in a primary melanoma with UV damaged skin? And if you look at the epidemiology, you see that some people with very chronically sun-damaged melanomas die more. Here's the risk of death. And some very sun-exposed sites are protected. And this is Australian data, just a breakdown. The American data also shows something very interesting, which is if you have a very sun-protected melanoma, you die more than if you have a melanoma arising either over chronically or intermittently sun-exposed sites. So it doesn't suggest that it's actually UV light that's killing our aged patients. So what's killing them? I looked directly at solar elastosis in the literature and found the Berwick study, which found a protective effect from solar elastosis. So more UV damage, more sun, actually decreases your risk of death with melanoma. These studies have not been validated in subsequent very important groups. And of course, there was this seminal Nature paper from Thomas Tüting, which showed that actually, when you give UV light to an animal model of melanoma, you have more metastasis and more death. So I approached this question by taking fibroblasts, which are, of course, the ones that are damaging the dermis, from German donors. This is head and neck and trunk donors across an age group. And I did RNA and DNA sequencing. And as the number of UV damage mutations, signature 7 once again increases, what we observe is, as expected, collagen degradation. So these are extracellular matrix catabolism enzymes with a loss of increasing anabolism of collagen. So you stop producing collagen and you degrade too much, which leads to your wrinkles and saggy skin. And the more you sun expose, the more you get. So to study this, I took human foreskin fibroblasts and I generated isogenic cell lines that are UV damaged. I did atomic force microscopy. As expected, all the fibers are broken. And this not only affected collagen, but multiple other fibers. About 300 proteins in the extracellular matrix are dramatically different with just 8 doses of mild UV. So a completely different matrix. We know from histology that collagen in blue varies across melanoma samples. I've correlated this to mutation damage. And you can see that the chronically sun-damaged mutation samples have fibroblasts with very high burden of mutations, as you'd expect, and the opposite is true for melanomas arising over intermittently or sun-protected sites. So my question was, what will happen if you have collagen degradation and a melanoma arises in this type of skin, pre-damaged? So the way I modeled this is by doing melanoma spheroids and embedding them in collagen matrices that had different proportions of collagen in them, so stiffer versus blebbier. And what I found was really interesting, which is that regardless of the background, the molecular driver in the melanoma cell line, I always had this bell-shaped curve. So very, very stiff matrices didn't allow the melanomas to squeeze, cells to squeeze through, and very, very blebby and unstructured matrices equally stop melanoma invasion. So there is a sweet spot of invasion regardless of your molecular background as a tumor cell. So this was very exciting, and this is how the single cells invade, and you can count the single cells and you see the same thing. Then I checked what would happen if you degrade the collagen in a given matrix, as happens in the skin, really. And for this, I did exactly the same experiment, same collagen concentration, but degraded the collagen by adding a collagenase, which is an enzyme that breaks it down. And of course, you decrease the invasion, which is what happens in aged skin. So for now, I'm telling you that old people die more, but their melanoma is less invasive. So I'll get to that in a bit. I took my model of cells from the human foreskin fibroblasts. You can see this across multiple constructs and cell lines. Always when you have UV damage, you stop invasion, and you dramatically decrease the changes, and this affects all of the proteins in the extracellular matrix. This is fibronectin. We mimic every single change that UV damage does in vitro. I took real patient fibroblasts this time from melanoma patients, and I looked at MMP1, which is the collagenase, and melanoma invasion. And as expected, the more MMP1 the fibroblasts chuck out from real patients, the less the melanoma invades. But what was really striking is that this relationship was much stronger and inverse with collagen expression. So the more collagen a tumor produced by the fibroblasts, the more able they were to invade. So what's happening in patients? I took a cohort of patients from many countries. These patients were over 55, and I scored the single-cell invasion, the solar elastosis, and the amount of collagen in the matrix. And this is just an example. So this is a melanoma invading in the dermis, and it's sort of blebbing out, or as an expansile nodule. Very few cells are singly detaching, compared to here, where all the cells seem to have acquired the ability to sift through the collagen fibers. So I call that high invasion versus low invasion. And I scored the solar elastosis, so perfect fibers and the blebby UV damaged skin. And so what happens with survival? So this is what happens. I found that the low invasive tumors, so very few cells detaching, have a really good outcome. And this is more robust than any of the current markers of survival. And the ones that have many cells singly invading through the fibers at the primary level die. This is inversely correlated to the amount of solar elastosis or sun damage, obviously, because you have less collagen, they invade less, exactly like in vitro. So I also checked what the correlation with survival was with a tumor-adjacent collagen degradation, so the real marker of sun damage we have. And curiously enough, it's a trend. The more sun damage you have, the better you do, but it's not significant. So I wondered, well, why is that? Why would be single-cell invasion so much better than the other one? So to address that, I decided to score the collagen directly at the invasive front of the tumor. So forget about the tumor-adjacent areas, because we know tumors have the inherent capacity to generate more collagen. So I looked at that, and then, lo and behold, I found a really, really strong correlation between more collagen at the invasive front, directly where the tumor is invading, and outcome. I'm going to show you a few pictures. This correlates with collagen expression in primary tumors, and it is extremely strong, and pan-cancer, may I say. This is just an example of a chronically sun-damaged melanoma. This is the sun-damaged degraded skin. You see the blobs, no collagen, and a very sharp transition between the tumor and the dermis. So no cells are detaching and invading. So I hypothesize that these are the guys that are doing well. And then, of course, you have these tumors with complete collagen degradation here. But you can see this band of new collagen synthesis that is bright pink. I'll show you a close-up. This is what it looks like. And you see that tumor cells are sifting and shifting through the collagen fibers. So I speculated that actually these are the guys who are dying. And yes, it's true. So the very rapid, I'm saying here it's less than 5-year survival, but it's closer to 2 years. If you have those collagen bands with new melanoma cell invasion, you actually die quite quickly when you're old. So that explains perhaps why they're dying in relationship to UV light. So these are the survivors and these are the dyers. Essentially what I've shown you is that melanoma, UV light will stop melanoma invasion unless, of course, you switch on collagen production. And we're currently working on mechanisms of why some tumors switch it on and others don't. And, of course, ways to avoid this. It's a very complicated UVR policy message. We're all saying stay away from the sun, but I'm telling you sun is good unless you make collagen. I just wanted to point out this really interesting study showing that people who sun exposed live better, and this is adjusted for comorbidities. So you cannot draw any policy conclusions from this. There'll be many, many other reasons why sunlight is important, not to mention vitamin D and systemic health. But there will be a safe range, and this will, of course, affect policy in the future by skin type. I'm going to talk a little bit about sex. As you can see here, the majority of people who are dying are old white men. It is currently the 4th biggest cancer killer in the UK for old white men. All my studies that I call aged are over 65. I've just drawn the line at 50 to drive the message home. So I was very interested, and I looked at mutations in cell division in melanoma tumor samples, just like I did for signature 7. Signature 1 mutations reflect that inherent mistake that cells make when they divide, so they always carry on a little mistake. And you can actually see that the older you are, the more cells, the more times your tumors have, your cells have divided, as you would expect. But what was very interesting is that we saw a dramatic difference by sex. So men seem to have more, men's melanocytes seem to have an inherent clock mutation or proliferation rate that is very different to women. And this has been shown across many other cancer types or differences by sex. Melanoma is a very expensive mouse model, so I decided to study the question in another very sex-biased tumor, which is squamous cell carcinoma, which is very cheap to model. This is what it looks like. It primarily affects men, and it primarily kills men when it does kill. I think the incidence in this country is 4 to 1 men to women. Everybody thinks it's because men are bald and have, more sun exposure. I strongly disagreed with that, or I wanted to challenge that hypothesis in view of my molecular data. I speculated that it was probably hormonal or other chromosomal causes. So to study that, I looked at a cohort of Salford St. George's Hospital as well, prevalence study, and I did see that was very striking, that men seem to have, for a given population of about 1,000 tumors, always a higher histological grade. But of course, this could reflect that they've had more sunlight exposure and they're just simply more aggressive or being diagnosed late. It was still very striking. I looked at advanced stage, and once again, I saw the same incidence. So men tended to have more aggressive disease, and there were far fewer females than men going on to have metastatic disease from cutaneous SCC. So to model the true role of sex, I took animals, I treated them to a very standard and cheap model of carcinogenesis, and I recorded what SCCs they would get. And actually, what I found is that the male animals were the only ones that were metastatic. So here you control age and you control dose of carcinogen. And the tumors were inherently more aggressive in a male background. They had more undifferentiated types of tumors, just like you see in your prevalence study, and more lung metastases. So my first hypothesis was, well, is this because with the same damage they're repairing fewer mutations to it? Do they have worse DNA repair than females? And they don't. They actually repair mutations at the same rate as females. So even though you have two tumors with the same amount of mutations, it's always worse for men. So the next thing I did to study that is I looked at the transcriptome of normal skin that has been exposed to the carcinogen, so the very early changes. And what was really, really striking was that females upregulated strong anti-cancer immunity pathways, whereas male animals did not. So the early response to damage was protection with immunity from females. And they actually had a much higher influx of cancer-fighting T cells in their tissues than males. Same dose of carcinogen. In contrast, men had a more proliferative rate at their epidermis that was damaged. So you see men proliferate more in response to damage and females upregulate immunity in the animals. So does this hold in humans? Well, sorry, I just crossed down the most important genes. The most upregulated one in females was CDKN2A, which is a tumor suppressor gene, which also links to the immune response. And actually, when you knock it down, it becomes more malignant in females. And I also gave my animals prednisolone to knock down the immune system and see if then female animals would have the same type of disease as men. And that's exactly what I saw. When you knock down immunity in females, their disease resembles male disease. So they lose their protective immunity and they tend to have very aggressive disease. This is human single-cell sequencing of epidermis, and we see exactly the same thing in adult human skin. So the female keratinocytes that have been UV damaged upregulate interferon gamma and many other pathways and immune cell landscape in keeping with with a tumor-fighting response. And this is not upregulated in men. And critically, when you take an immunosuppressed population of UK patients, what we see is that females that are immunosuppressed suddenly have equal amount of disease to men. So that's the great equalizer for both. That's it. I wanted to just highlight the role of sex and age in skin cancer. Thank you very much. I have so many people to thank. They're all there, especially my funders.